everyone, thank you for coming to my talk on spatial deposition and larynx developmental analysis of 6-year-olds through CFPD. It's been well established that there are variations between adult and pediatric airways in their geometry and anatomy. Specifically for us, we're interested in the glottis being the narrowest point for adults and the cricoid ring being the narrowest point for children, amongst other geometry variations. And there's a need for models that represent pediatric airways for disease targeting and also to avoid upper throat deposition for efficient lung delivery. But there's lack of models which quantify this transition between the pediatric-like to adult-like airways. And since this transition occurs in 6-year-olds, the goal of our project was to create and analyze reconstructed mouth-throat airways of 6-year-old subjects to study this transition, as well as to identify key parameters and aerosol deposition in these models and compare to the pediatric Alberta idealized throat, which is a commonly used idealized model that represents ages 6 to 14, but it was based on the geometry of an adult model. So we're hoping to determine where it does or doesn't predict aerosol behavior well during this anatomy change. And finally, once we have a better understanding of how these models behave fundamentally, we'd like to utilize this in a practical study using a realistic drug model based on the advert discus. And we hypothesized that the pediatric-like anatomic features would noticeably influence the mouth, throat, and downstream pulmonary deposition when compared to models with more adult-like features. And we worked with the clinician, Dr. Jenna Burdell, who annotated CT scans such that when we 3D rendered the airways, we were able to correlate to anatomic features of interest, such as the nasal-oral pharynx through the trachea. And we did this for 2 CT scans of 6-year-old subjects, 1 male and 1 female, and as well as the Alberta idealized throat. And we used a combination of in-house and open source softwares to quantify a center line through this model. And the center line is shown here in black dots, and the red dots are deposited particles from CFPD. So instead of quantifying deposition along an axis, we quantified deposition between 2 points of the center line. And so that's what we're seeing down here. This is the distance along the center line, and the average deposition fraction is representative of particles that deposited between 2 points of the center line as we traveled along it. So then we used other metrics along with this, such as model diameter, surface area, angles through the models, etc., to quantify anatomical features of interest. And we did this for all 3 of the models that we used. And we can see, for example, that the glottis of the Alberta idealized throat is more narrow than the cricoid ring area. And we use this type of analysis to quantify the anatomic features and determine if they were more pediatric-like or adult-like. So, for example, that Alberta idealized throat, the narrowest feature was at the glottis, so that was more adult-like, and the Alberta idealized throat has predominantly adult-like features. Subject A had a mixture of adult and pediatric-like, and subject B had predominantly pediatric-like features. And we expected that to translate into fluid dynamic metrics as well, such as the relative turbulence measured through turbulent intensity, recirculation regions, quantified by comparing the axial and tangential velocities, as well as the Reynolds number through these models. And we quantified these metrics for all 3 models, the Alberta idealized throat, subject A, and subject B. And we saw that there was more turbulence, recirculations, and higher Reynolds number in the airways with more pediatric-like features. We saw that the Alberta idealized throat and subject A had entirely adult-like features, while subject A had entirely pediatric-like features. And this is qualitatively what we would expect based on literature. And again, while this is qualitative, while this is qualitative, we did find that these are interesting comparisons that could be useful in future differentiating between adult-like and pediatric-like airways. So now we have successfully created these 6-year-old models and quantified their anatomical features, and we'd like to better understand which of these features are key in predicting aerosol deposition and how they compare to the pediatric Alberta idealized throat. So beginning with particle diameter, we quantified the deposition fraction along the center line for the 3 models, the Alberta idealized throat, subject A, and subject B, as we increased particles from 100 nanometers to 10 micrometers in diameter. And we found that there was significantly less deposition with smaller particles than with larger particles. And there was a noticeable difference between sizes 1 micron and 3 micron. And and greater. And this was true for all three models as well. So we can say that deposition fraction varies significantly with particle size, and that there's a noticeable increase in deposition for particles that are greater than or equal to 3 micrometers. And in a similar comparison for flow rates, we can see that there is less obvious variation as we increase the flow rate from 10 liters per minute to 120 liters per minute, and that trend holds true for subject A and subject B as well. And so we can say that the deposition fraction does not vary significantly with flow rate. But it's important to note here that the Alberta idealized throat accurately predicted those trends. And there is also a difference in where these particles are depositing. So for example, for the Alberta idealized throat, there's more deposition in the supraglottis. For subject A, there's more deposition in nasopharynx, and for subject B, there's a combination between the two. And along with those kinds of trends, we also looked at trends regarding where particles tend to deposit or hotspots in these models. And we are continuing to analyze those types of trends, but I'm not going to go into that in more detail today. Besides those individual trends of diameter and flow rate, it's easy to visualize the comparison between these models using a graph of d squared q versus the fraction of deposition. So if we look at the Alberta idealized throat as squares and subject A as circles, we can see that there is overlap between those two models, more so than there is in comparison to subject B, which has significantly more variation. And we can also see this if we separate out by particle size in each of these panels, as well as by flow rate, where the increasing, the darkening of these symbols correlates to increasing flow rate. We can see that there is more overlap between subject A and the Alberta idealized throat, and even where there is some overlap with subject B, the trends in those cases are significantly different. So we can say that the Alberta idealized throat is better at predicting an adult-like throat than a pediatric-like throat for this metric. So now we have a fundamental understanding of the influence of these adult-like and pediatric-like features, and we'd like to assess a realistic drug as a practical application for this work. So we did that by adjusting the particle diameter and the flow rate of these models. So instead of just having a set diameter, we varied the particle diameter to match the distribution of advar particles, and we adjusted the flow rate to be what is required to actuate the advar discus. And we compared our results to literature for the pediatric Alberta idealized throat on a next generation impactor with an advar discus, and found that the Alberta idealized throat better predicted the results of subject A, especially in the MMAD, compared to subject B. So again, this was the Alberta idealized throat better predicting a more adult-like throat, even though there were some other variations here as well. So to summarize, we created models for 6-year-old throats to quantify and study aerosol deposition patterns in the transition between pediatric-like to adult-like geometries. We identified key parameters of interest and metrics for which the Alberta idealized throat does or doesn't predict trends well. And notably, the Alberta idealized throat tends to better predict deposition in a more adult-like throat. And there were notable limitations to this study, and we hope to improve on this work as we expand the set of models used to explore variations that we've observed so far. And with that, I'd like to thank my lab, my collaborators, and my funding. I'd like to thank you for listening, and I'd be happy to take any questions. Thank you.